Hi, my name is Alyssa, and in this video we are going to examine the molecules and processes through which different tissue types derive their energy during the metabolic states. We'll start off by looking at what molecules are metabolized at each of the different tissue sites. Then, we'll examine specifically how these molecules are broken down to provide energy to the tissues listed below. After watching this video, you will be able to recall specifically what is happening in a tissue during the different metabolic states. You will be able to answer questions that ask you to think about which molecules are used in metabolism at the different tissue sites. First, let's explore the molecules specific to the metabolism of different tissues during two different metabolic states. This table lists the major sites of metabolic activity in the body: the liver, skeletal and cardiac muscles, adipocytes, and brain tissue. We add red blood cells here because, unlike other blood cells in the body, they participate in the Cori cycle, the cycling of lactate between red blood cells and the liver. In the liver, glucose and amino acids are the primary source of energy during the well-fed state, but when food supplies are low, the liver uses excess fatty acids for energy. At rest, skeletal muscles will use glucose as a source of energy when these stores are high. However, during the fasting state, fatty acids and ketones become the major source of energy. In cardiac muscle, fatty acids are metabolized during both the well-fed and the fasting state, but during periods of fasting, ketones can also be used for energy. Adipose tissue will metabolize glucose in the well-fed state and fatty acids in the fasting state, while the brain can really only metabolize glucose in both states. However, after over 1 week of fasting, the brain begins to use ketones as an energy source. Red blood cells can only use glucose for energy, even when glucose stores are low during the fasting state. And that is why it is important for other tissues to rely on other molecules for energy during this metabolic state. The remainder of this video will investigate the metabolism of most of these tissues in detail. We'll start with the liver. The liver has two main roles in fuel metabolism. The first is to maintain a constant level of blood glucose during all metabolic states. Let's go through this in detail. Before eating a meal, the concentration of glucose in the portal vein is low to normal. Remember that the portal vein is where nutrients from the intestine are absorbed so that they travel directly to the liver. After the meal is complete, proteins, fats, and glucose that were ingested and absorbed from the GI tract travel through the portal vein. At this time, the concentration of glucose increases. Therefore, it is time for the liver to perform one of its main roles in metabolism. Let's take a look at what happens next. After a meal, glucose concentration in the portal vein is high. This stimulates the beta cells within the pancreas to release insulin into the bloodstream. Once these levels are increased and insulin reaches the liver, the anabolic processes begin in order to decrease the concentration of glucose. In the liver, insulin promotes glycolysis, which breaks down glucose into pyruvate, which is used for the synthesis of acetyl-CoA. It also stimulates glycogen synthesis, the formation of our body's storage medium for glucose, which will decrease the concentration of glucose in the bloodstream. Insulin also promotes fatty acid synthesis, so let's take a brief look at that. In glycogen synthesis, not all of the glucose can be converted to glycogen, and excess glucose is instead converted into acetyl-CoA. This molecule is then used in the synthesis of fatty acids within the liver. Once synthesized, 3 fatty acids undergo an esterification reaction and bond to a molecule of glycerol to make a triacylglycerol molecule. These molecules are then released into the bloodstream as very low-density lipoproteins, molecules used for energy when glucose is unavailable. So let's now take a look at the metabolism within the liver during a period of fasting and how it then maintains blood glucose levels. In a fasting state, blood glucose levels are lowered. This stimulates the pancreatic alpha cells to release glucagon into the bloodstream. Glucagon travels to the liver where it stimulates the catabolic processes that will break down molecules into glucose, thereby increasing blood glucose concentration. These processes are glycogenolysis, the production of glucose from glycogen, and gluconeogenesis, the synthesis of glucose from other molecules. These processes act to increase the concentration of glucose in the blood. Let's take a closer look at gluconeogenesis and where the carbon atoms required to make glucose come from. The first molecule is lactate, which is carried in the Cori cycle from red blood cells to the liver. The second molecule is glycerol from triacylglycerol molecules, and finally, amino acids from the breakdown of proteins found in muscle tissue. These molecules combine to form glucose in the process of gluconeogenesis. However, this process takes about 12 hours to reach its maximum rate, whereas glycogenolysis begins right away.
The second major role of the liver in fuel metabolism is to synthesize ketones through the beta-oxidation of excess fatty acids during periods of prolonged fasting.
The importance of synthesizing these ketone bodies is that they provide an alternate source of energy to glucose. Glucose synthesis requires amino acids to be catabolized, but by using ketone bodies, these amino acids can be spared for other essential functions within the body. These ketone bodies are easily transported through the bloodstream and can absorb into cells. Therefore, they are considered transportable forms of acetyl-CoA because they can go anywhere and provide energy to most tissues.
Let's now investigate the process of synthesizing these ketone bodies.
During the fasting state, the liver is oxidizing fatty acids into acetyl-CoA. In a prolonged fasting state, this oxidation process is increased, and therefore levels of acetyl-CoA are increased. This drives the process of ketogenesis, which occurs in the mitochondrial matrix and begins by producing HMG-CoA and eventually acetoacetate, a ketone body. Acetoacetate can be further reacted to form 3-hydroxybutyrate, and these two ketone bodies can then be used for energy throughout the body. Acetone is also made in this process, but it is excreted through the urine. Ketone bodies are released from the liver so that they can serve as an alternate fuel source for most tissues in the body, except for the liver and red blood cells. But remember that ketone bodies will only be used for energy in the brain after several weeks of prolonged fasting. The liver cannot use ketones for fuel because it lacks the enzyme thiophorease, which is required to catabolize them. This makes sense because these compounds are synthesized in the liver, and they wouldn't be able to reach other tissues if they were immediately metabolized for energy in the liver. Let's take a minute to think about what the preferred fuels are for the liver during the well-fed state.
We saw how glucose is used to synthesize molecules used for energy storage.
It can also be used right on the spot to provide energy for the liver. But what we didn't look at is that the liver derives most of its energy from the oxidation of excess amino acids. However, in times of fasting, these fuels change so that the essential amino acids are not degraded to promote the processes of decreasing glucose concentration.
We just saw that in a prolonged fasting state, when glucose levels are low,
the liver uses fatty acid oxidation, which synthesizes acetyl-CoA for energy. Now let's take a look at what happens in adipose tissue during the well-fed and fasting states. During the well-fed state, insulin levels are high, and this activates the enzyme lipoprotein lipase. This enzyme promotes the cleavage reaction that leads to the release of fatty acids from stored lipoproteins. These fatty acids are then taken up into the adipocyte and undergo an esterification reaction with glycerol to yield triacylglycerols for storage. The more triacylglycerols stored, the larger the cell will become and the more energy reserves it has for use during later metabolic states. During the fasting state, insulin levels decrease and glucagon and epinephrine levels increase. These hormones activate hormone-sensitive lipase, which hydrolyzes the fatty acids from the stored triacylglycerols and releases them from the adipocytes. Let's see what this process looks like. Without these hormones, hormone-sensitive lipase is inactive and triacylglycerols are stored and not broken down.
However, in the fasting state when epinephrine levels are high, epinephrine binds to an external receptor on the adipocyte, and this activates the enzyme.
Then, stored triacylglycerols are broken down into fatty acids and glycerol. These molecules are then released from the cell, and the adipocyte shrinks back down to the size it was before storing the triacylglycerol molecules. The released fatty acids travel through the bloodstream attached to serum albumin and end up in the peripheral tissues to be used for energy.
Glycerol then travels to the liver, where it is then used in the process of gluconeogenesis.
Let's move on to skeletal muscle and see how this tissue type performs during the different metabolic states. During the well-fed state, increased insulin levels cause the muscle tissue to take up glucose molecules into the cell. Insulin binds to an external insulin receptor on the skeletal muscle,
causing signal transduction, which stimulates the movement of additional glucose transporters to the membrane by a mechanism involving exocytosis of vesicles containing these transporters. With increased glucose transporters on the membrane of the muscle cells, glucose in the bloodstream can then enter the cells within the tissue and be used for energy.
These molecules can also be used to synthesize glycogen to replenish the molecules that were used previously for the muscle's energy requirements. In resting muscle tissue, glucose is the primary source of energy while in the well-fed metabolic state. During the fasting state, glycogen stores are depleted, so resting muscle uses fatty acids circulating in the bloodstream for its energy requirements. If fasting has been going on for a while, ketone bodies can also be used to provide energy to the tissue. There's a difference between the substrates used for energy for resting skeletal muscle and for active skeletal muscle. The type of substrate being used depends on the muscle fiber being used and the type of exercise being performed.
First, we'll look at the fuel sources used depending on the duration and intensity of the exercise. This information can be organized into a table to help you recall it on test day.
For short 2 to 7 second durations of intense exercise, the fuel source is creatine phosphate. Intense exercise of either seconds to minutes uses stored glycogen in anaerobic glycolysis. For less intense, more moderate exercise of continuous activity, glucose and fatty acids are used as a source of energy for the muscles. And finally, for continuous use over 1 hour, the intensity will begin to decline as the activity continues, and once all of the glucose has been used, fatty acids act as the fuel source. So we've looked at the duration and intensity. Now let's focus on the muscle type. For short, intense exercise, the fast-twitch muscle fibers are used. These fibers are quick to fatigue, but function well for a short time on anaerobic glycolysis. For longer, more moderate exercise, the slow-twitch fibers are used, which contain a lot more mitochondria than the fast-twitch muscle fibers, and therefore use oxidative phosphorylation to make ATP.
Make sure that you are familiar with the two muscle types shown, and that you can recall what the fuel sources are during the different types of exercise.
In cardiac tissue, the preferred fuel source is fatty acids. In the well-fed and in the fasting states, fatty acids are used as the primary fuel source. It is only after periods of prolonged fasting that fatty acids share their responsibilities with ketone bodies, and both these types of molecules are used for energy. Though ketone bodies can be used at this time, fatty acids are still the preferred fuel. The brain is a complex tissue, and though it is small compared to the rest of the body, it consumes 25% of the body's total glucose supplies. This supply of glucose to the brain must be regulated so that the brain continues to function even when glucose supplies are low throughout the body. During the well-fed metabolic state, glucose passes through the blood-brain barrier and is used as the primary fuel source in the brain. Uptake of glucose into the brain is insulin independent, and during periods of fasting, glucose is supplied to the brain through the bloodstream from glycogenolysis or gluconeogenesis that occurs in the liver. Fatty acids are unable to cross the blood-brain barrier, and therefore cannot be used as fuel during any metabolic state, no matter how low glucose concentrations are.
After periods of prolonged fasting, glucose concentrations are low, and the brain's primary fuel source is unavailable. The brain then needs to find an alternate fuel source.
Since fatty acids cannot cross the blood-brain barrier, ketone bodies act as this fuel source, as they can cross the barrier and act as an alternate source of acetyl-CoA. Though the brain prefers glucose, it can derive 66% of the energy it requires from the metabolism of ketone bodies. Now that we have explored a number of different tissue types and how they obtain energy, let's try out some practice questions to test your understanding of the topic. This first question is asking you to think about each tissue and its preferred fuel source. The answer is D, cardiac myocytes. A is incorrect because skeletal muscle utilizes glucose in the resting state, as well as muscle glycogen. It is correct that skeletal muscle uses fatty acids as a fuel source. However, it does so during prolonged moderate exercise or in the fasting state, not in the well-fed state. B is incorrect because hepatocytes utilize glucose and amino acids during the well-fed state, though they too can use fatty acids during the fasting state. C is incorrect because adipocytes also utilize glucose in the well-fed state. However, these cells do take in fatty acids during the well-fed state, but that is done for the synthesis of triacylglycerol molecules and not for their utilization for energy. This final question is asking you to think about the specific aspects of each tissue's metabolic processes. The answer is A. The liver does not have the required enzyme to metabolize ketone bodies, though this is the tissue type responsible for synthesizing them. Answer choices B, C, and D are all correct statements. Great job if you got those questions correct. In this video, we reviewed the different molecules used in metabolism in various tissues and saw how these tissues undergo metabolism during the different metabolic states. We saw the different tasks that the liver is responsible for, being the most metabolically diverse tissue. It maintains blood glucose concentrations through gluconeogenesis and glycogenolysis, and produces ketone bodies to be used for energy in the same way as acetyl-CoA. We saw that adipose tissue stores lipids when insulin levels are increased, and releases them to be used for energy when epinephrine levels are high. In skeletal muscle, active muscle uses different fuel sources depending on the type of exercise being performed, and resting muscle uses glucose to make stored glycogen while using fatty acids during the fasting state.
In cardiac muscle, we saw that fatty acids were the preferred fuel source during all metabolic states.
Finally, in the brain, glucose is the primary fuel source, even during fasting. However, after prolonged periods of fasting, 2/3 of the brain's energy can come from ketone bodies. Thank you for watching this video, and good luck studying for your MCAT.